Welcome, everybody. Today we're talking about methylation, which has been a popular subject in the past number of years due to the genetic sequencing that we can now do. However, there's a lot of things about methylation that people don't know, like how much of this actually has an impact on one's health. And so we're doing all the sequencing, these genetic tests that tell us we need to intake more folic acid or folate. But there's a lot behind it. And there's a lot of ways that we can look at not only methylation, but the whole of biology to understand what we can do to best impact our health. So this is just one piece of the puzzle, methylation. And so we're going to be putting that into the context of where it fits into biology, how big of an impact it has, and how you can better just understand it. So today we're talking with Dr. Jasbi, who is an expert. He was a researcher at Arizona State University and now is the CSO and co-founder of a company called Theriome. Theriome specializes in metabolomic testing and systems biology. Well, what does that mean? Genetics are a part of systems biology, so they are at the top. That's our nature, what we are hardwired to do, what we are predisposed to potentially have in those various things. But there are other levels of our systems biology that are very important as well, such as the transcriptome, the proteome, which would be like our proteins, hormones, all that kind of stuff. The microbiome, many people familiar with, and if you watch other videos, you are familiar with Dr. Alex Moore, who teaches on the microbiome. So Dr. Jasbi is an expert in systems biology, having spent many, many years in research and has published quite extensively on systems biology and just the impact that has for various things, be that early detection or improving people's lives. So thanks for joining today.
Absolutely, Mike. I'm so thrilled to be discussing this topic. It's one that is very much in vogue and unfortunately very much misunderstood. So I'm looking forward to diving into the presentation today and hopefully educating and letting people know what these tests are and what they're not. Yeah, as Mike mentioned, my name is Paniz Jasbi. I received my PhD in Arizona State University's College of Health Solutions. I completed my postdoctoral training at ASU as well, in a different college, the School of Molecular Sciences, and I currently serve as the Chief Science Officer of Theriome. Thanks for joining us today, and if you're watching this, thanks for dropping in later. So we're going to be talking about methylation and doing a quick compare and contrast with our in-house test, our flagship product, the Aristotle test. Our work essentially is dedicated to pioneering the frontier of health testing through a very comprehensive understanding of the human body's myriad complex systems. So traditional genetic tests, now this is not whole genome sequencing, but traditional tests, including those that examine the methylation-related genes. There's four major ones that we'll be discussing today. MTHFR, this very notorious gene that everyone is very concerned about, ComT, AHCY, and MTR. And these are genetic markers that will undoubtedly offer valuable insights, but they truly, as we'll get into, really just represent a fragment of the intricate puzzle that is human health. So today we'll introduce you hopefully to a more holistic approach. And there are very holistic tests on the market besides our own. Our test is known as the Aristotle test, named after the philosopher who first committed to writing a formal theory of metabolism. And it's a cutting-edge analysis that aims to transcend the boundaries by integrating systems biology level knowledge and metabolomics. And we'll get into what that is. And the goal with this test is to offer you a panoramic view of your health, a really unprecedented probing of your cellular physiology. So let's embark on today's educational journey. And hopefully by the end, we will have redefined what it means to understand your health. This is what methylation is at its core. The addition of one group, right? So we go from cytosine to 5-methylcytosine. So it's bound to the 5th carbon here, a methyl group. And that is essentially what it is. And so we're going to focus on 4 methylation genes that are very important. And those are MTHFR, MTR, AHCY, ComT. We'll discuss what they are and what they're not. It's really best known for affecting DNA expression and function. So if we methylate cytosine, we can silence or indeed induce the expression of certain genes by methylating their base pairs. That is something that we can do, right? And so it's really known for DNA expression, but we will talk about it today in the context of its cellular biochemistry, which is estrogen and neurotransmitter synthesis, and then again folate, B12, and homocysteine regeneration. And so that is what it really does, and it does have some effect on DNA expression, and of course myelination. So in the central nervous system, we have
That's what I'm thinking,
oligodendrocytes.
myelination, right?
Yeah, absolutely.
Yeah.
You got it right. So in the central nervous system, we have cells called oligodendrocytes, which take part in methylation processes to form the myelin sheath on the axons of neurons, and that allows for unimpeded transduction. Essentially allows electrons to jump from node to node. Those are known as nodes of Ranvier. And the same thing happens in the peripheral nervous system by a different cell type known as Schwann cells. So you're absolutely right. That is one thing that methylation is known for. And we'll see that they're known for many things. But at its core, it is the addition of a carbon bound to three hydrogens. So they offer insights, no doubt, but they're limited. They have a narrow focus, and they can be potentially misinterpreted, and often are misinterpreted without the broader context. So we're going to first be discussing ComT. So this is a gene, right? So when we talk about these genes, you should know that there's a gene that you have, and you have different variations of these genes. And then we have the RNA. So those genes are then transcribed to RNA. And so the DNA code is made into RNA code. And then those RNA go into cellular machinery known as ribosomes. And those ribosomes are then reading amino acids and folding them into proteins. Think of them as protein synthesis factories, right? So now we have translation from RNA to protein. And then those proteins go and interact with other proteins and metabolites and gut microbiota, et cetera. The metabolites interact with gut microbiota, et cetera. And they produce metabolites and have other various downstream effects, right? So when we talk about the central dogma of biology, from expression of the nuclear DNA down through the metabolites, this is what we're talking about. So there's a gene for every protein, right? There is a ComT gene which controls the expression of catechol O methyltransferase, which, as you guessed it, transfers a methyl group. Now, this is a fairly ubiquitous enzyme. This enzyme, which is a protein, enzymes are proteins, again coded by the ComT gene, is really involved in the breakdown of neurotransmitters, hormones, and various drugs. Okay, so variations in this gene that you and I have variations of, these influence the metabolism of estrogen and neurotransmitters, and they can impact many domains. They can impact mental health. They're implicated in pain perception, and given the number of substrates and products that they act on and produce, they are implicated in numerous health diseases. So what does it do? Well, as I mentioned, neurotransmitters and estrogens, these are big parts of it. So the conversion of dopamine to 3-methoxytyramine, this is controlled by ComT. A derivative of dopamine, DOPAC, to homovalinic acid, or HVA. This is controlled by ComT. Norepinephrine to norepinephrine. ComT is responsible for that. This methyltransferase enzyme that's coded by the ComT gene. Epinephrine to metanephrine. Dihydroxyphenylethylene glycol to methoxyhydroxyphenyl glycol. Dopeg to mopeg. And then finally, we have this DOMA to VMA, this vanillomandelic acid. So these are very important substrates and products, and they are regulated by the ComT gene. So depending on the variation you have, your regulation in these may be greater or lesser or non-existent at all, although that's very rare. And that is what I think a lot of people confuse, is that I have, let's say ComT or MTHFR mutation. That means I don't produce any homovanillic acid. No, it usually means that you produce less than, or sometimes it can mean that you produce more than, but hardly ever not at all.
And then what are the kind of the practical implications there? So, I mean, these are chemical processes that are happening, but if you are not expressing the ComT correctly, or you have a mutation there. What are kind of some of the practical implications of what may happen?
Great question. So let's take the first one, dopamine to methoxytyramine. So this is a process when we have enhanced dopamine metabolism, particularly in the part of brain known as the substantia nigra, where the black substance relies. That starts to turn grayish white. It goes from a black area to a grayish white area. It's black because there's a concentration of dopaminergic neurons there, or neurons that produce dopamine. And so when we have this gradual ablation in those dopamine producing neurons because of an overactivity of ComT, what we get is, we induce essentially, is Parkinson's disease. So this is one of the conditions that is related to ComT. And in fact, when you give, when you give levodopa, and then you give medications to reduce, levodopa degradation, and then you also follow it with a inhibitor of ComT activity. That's known as the big three cocktail for Parkinson's disease, and has become the standard of care. So definitely ComT has important functions. For instance, if you're not producing enough norepinephrine or metapinephrine, you're going to have a deficit in energy levels. If you're producing too much HVA, you're going to have, or not enough HVA, I should say, you're going to have depression, most likely. It's one of those molecules that's very concomitant with depression. So as I mentioned, most of these are mental health issues. Most of these are issues related to pain perception. And then some certain diseases like Parkinson's disease. Absolutely.
Okay. And then what's like the significance of it though? Like how large of an impact? So we know that there's nature, which is what our body is hardwired to do. And then there's nurture, which is all our lifestyle decisions and that kind of thing. Do we have a pretty decent understanding of it? I know it's not just ComT and we're kind of getting into it, right? There's the three other genes that we're going to be talking about that are very important in methylation, but what is the significance of this, and how likelihoods and that kind of thing?
Yeah, great question. So there are obviously some genes that are more important than other genes, but we will get into this further. But there are roughly 200 genes that we know of that are responsible for facilitating methylation. So if methylation as a general process is incredibly important, we're only looking at 2% of them with these genetic tests that zero in on these 4 genes and sort of make much ado about ultimately probably nothing. So if I'm talking about percentage wise, I would say less than maybe 0.1% of overall health. 0.1% of overall health is the variance is explained for by variations in this ComT gene.
Yeah, and just for further clarifying context, which I think we covered, but the reason we're focusing on these 4 genes is because these are often used in genetic tests as kind of the 4 staples for methylation. But I guess my other question then is, so why did they grab, or why do a lot of groups grab these 4 methylation genes? If I had to speculate, I would wonder if it's because they're looking at people with Parkinson's or various illnesses and saying, okay, all people with Parkinson's, or at least a good majority of them, have these 4 genes that are mutated. Like, do you know, that's a speculation, but do you know why these 4 might be actually utilized in these tests so broadly?
Sure. I think it's because of my personal opinion is because it's easy to test. So this is not a whole genome sequence. This is not an in-depth read of all the base pairs of the body. It does not need next generation platforms. They're relatively cheap tests, and it's easy to sort of wind a story around them. It is easy for these testing entities to come along and say, well, we have a story around these 4. And the other 3 really interact with each other, as we'll see in the following pathway. But there are, of course, astute physicians who, when someone comes in with, let's say a Parkinsonian, cluster of symptoms, they will test for ComT, and that's a very directed, targeted testing. And those are, trained physicians in personalized medicine. And most of these individuals, and precision medicine isn't yet a formal specialty of medicine, but there are those who've taken some education and do practice it the best that it's nascently available currently. And we have these these tests that are, as we'll say, important in methylation of B12. They're important in micronutrient metabolism, but they are very much, overrated, and they don't play nearly as important a role as we'll say. So they are easy to weave a narrative around. They are easy to sort of get people to understand. When we talk about the complexity of systems biology, it's vast. And one of the greatest challenges we have at Therium is translating that into a coherent, actionable, narrative for our customers. And so if we only had 4 genes regulating, 4 enzymes, that would be much easier. And it would be cheaper. And it would be sort of very much directed around the metabolism of these enzymes. And so that is why I speculate, Therium has aspirations of its own to do genetic testing eventually, but we will, of course, opt for WGS or whole genome sequencing to get the whole story. Because as we'll see, methylation is actually the least important chemical reaction. If we look at the total amount of chemical reactions that take place in the body and which type of chemical reaction is which type, methylation is the least important. And there's still 200 genes that we know of, roughly, as a round estimate, that facilitate methylation. And so even from that, statistic, it should be clear that these genes are only a very small story, a very small piece of a very large and much more intricate puzzle.
Yeah, and there would be some context-dependent information on that too, right? Like if somebody does have Parkinson's, then methylation will have a higher factor. Again, this is more of a question than a statement, but methylation would have higher of a factor.
In some instances, as you mentioned, the presence or absence of certain health conditions or predispositions based on familial risk might make the investigation of these genes more or less important for that person. We are talking about, sort of by and large, what is important and how much attention should be paid to it. When we talk about the other 3 genes, so this is a pathway where we see folate metabolism, B12 metabolism, and the metabolism of methionine down through cystathionine. And so we're going to be talking about these 3 other genes. So MTHFR codes for a enzyme known as methylene THF reductase. Okay, so the MTHFR gene, here we can see it, is responsible for coding. Let me put my laser pointer on. It's responsible for coding this 5,10-methylene THF to 5-methyl-THF. This is the function of this gene, is to code for this protein or this enzyme. Enzymes are proteins again, right? That takes this 5,10-methylene to 5-methyl-THF. Right? Now, this is important because the 5-methyl-THF is the form of folate that is responsible for methylating cobalamin to methylcobalamin. So if we don't get to 5-methyl-THF, if this gene is in some way compromised, then our methylation of cobalamin is also compromised. And if methylcobalamin is compromised, then it cannot then donate its own methyl group to homocysteine via this enzyme known as methionine synthase to derive methionine. And the elevation of homocysteine has numerous health effects. One of the most important being significantly increased risk for cardiovascular disease. So that brings us to our second enzyme, MTR, which is responsible for coding the enzyme or protein methionine synthase. So MTHFR codes for methylene THF reductase, which again produces 5-methyl-THF from 5,10-methylene THF. And this is important because it's this chemical reaction that creates a methyl group that cobalamin then comes along and says, I'm going to grab your methyl group. Now I have methylcobalamin, right? And then that methylcobalamin needs to donate its methyl group to homocysteine to create methionine. And in the process, it's regenerated back to cobalamin. And the enzyme that's responsible for doing that is MTR. And I love this pathway. This pathway is one of the most elegant clockworks in biochemistry, in my opinion. And so this is 2 enzymes, 2 genes that code for 2 enzymes that work in concert to do what? To use cobalamin as a methyl donor pool to grab folate methyl groups and donate it to homocysteine to alleviate that to lower that homocysteine level, right? So now we have 2 of these enzymes explained, okay? Now, After homocysteine is methylated, accepts the methyl group from methylcobalamin, we create methionine again. Cobalamin is regenerated. It's going to go back into that cycle and grab another methyl group from 5-methyl-THF, right? Then methionine goes to various other important molecules like SAM, and then SAM goes to acetinosyl homocysteine. Now we have this other enzyme that is much tested. This is AHCY. AHCY is responsible for taking acetinosyl homocysteine and generating homocysteine. What it does is it adds water, right? So acetinosyl homocysteine plus water equals what? Adenosine and homocysteine. So it creates two of these molecules. So this gene, AHCY, codes for this enzyme, adenosyl homocysteinase, which again is responsible for creating adenosine and homocysteine from acetinosyl homocysteine. So these are the genes. So ComT kind of works on its own in different pathways. It's involved in numerous substrate and product generation. But these genes are really just involved in the generation of 1, 2, 3, 4, 5, 6, 7. And so together there are 19 products here that we have, 19 substrates and products that are of the function of these 4 genes. So a lot of people say, oh, Dr. Jasbi, I have an MTHFR mutation, as if it's the end of the world and they're metabolically totally aberrant. That's not the case.
Put the horse down.
Yeah, exactly. Well, we got to take you out back now. Yeah, no, not at all. That just means that your folate metabolism may be at a lower throughput. It is much less likely that your MTHFR gene has a variant that it just doesn't work.
Yeah, throughput is the key word there.
Exactly, Micah. So it's not that you're not producing 5-methyl-THF and you can't, methylate your cobalamin and therefore your homocysteine levels are high and you're at serious risk for cardiovascular disease and your neurotransmitters don't work and you're going to get cancers and Alzheimer's. No, not at all. This is just one enzyme that is related to the metabolism of one nutrient. Now, that nutrient is linked to another nutrient, which is linked to a very important molecule whose levels we want to keep low, right? And so you may have a mutation in your AHCY that actually upregulates this throughput from acetinosyl homocysteine to homocysteine, which is bad, right? Now, can you have a confluence of factors whereby your MTHFR is low, your methionine synthase is low, and your AHCY is high? Yeah, of course. But there are easy ways to ameliorate that. And it does not mean by any stretch of the imagination that you're not producing 5-methyl-THF, that you're not producing methionine, or that you're producing too much homocysteine, okay? The chances are that the variation you have has lowered your throughput, has not completely obviated it, has not completely removed your ability to create these important intermediaries. It's just that you're doing them at a different rate.
What segment of the population would you suppose has mutations in these?
It's pretty high, actually. So, but everything can be high. There are people who have, 70% of the population has a SVCT1 mutation, which is regarded for the metabolism of vitamin C and other antioxidants. About 40% roughly of the population does have a mutation, MTHFR, but that 40% mutation shouldn't shock you at all. Because most of these genes that we'll talk about, that we'll say and see in these other slides, they also have mutations that are very prevalent among the population, more than 50, 60, 70%. But it doesn't mean that mutation is crippling or that there aren't other ways that your body is able to reach homeostasis.
Well, I think this is an important point that it may be. But so 40% has MTHFR mutation.
Sure.
What percentage of people would have one of these 4 that's mutated?
What do you mean, one of these 4?
Yeah, one
Probably
of these
pretty
4.
high. We all have different. What does a mutation mean? Mutation means just a variation. It doesn't mean that it means that we have like this one, standard type of gene that's coded, and then we have some changes in them. Do you think all of our genes are the same? Absolutely not. That's what makes us different. Right? If we had all these genes and there was no mutations in them, we'd all look like Micah or all look like me, or et cetera.
Yeah.
And metabolically, it's the same thing. So what are the chances that somebody has a mutation in all of these? Yeah, it could be as much as 20%. I really haven't
one
looked
of
at the
these.
literature. or
Because
it has
that's the
all
point I'm
four of
getting
these. I
back
mean,
to.
sorry,
It's like
all four of
why would
these.
somebody sequence these four in particular? And in my mind, if like over 90% of people have a mutation in one of these four, then that's a pretty good fit for saying like, hey, you have a problem that I can solve. If over
Okay,
90% of people have that problem that you test.
absolutely, but
I'm not
again,
sure if that's the case, but I'm just asking because I'm trying to vet this out and understand.
it may be the case that 90% of people have a mutation in one of these, or, as I kind of threw out, shooting from the hip, maybe 20% of people have a mutation in each one of these. That may be the case, but what does mutation mean? It just means that you have a variation. It could be that you have a mutation that's actually increasing the throughput of this. That's great. Those are beneficial mutations. That is how biology and natural selection passes on beneficial genes, is you have a mutation. It is analyzed in the context of how much it increases your likelihood of survival. And if it increases your likelihood of survival, it is more likely to be passed on to the next generation. So that mutation over time just becomes more prevalent in the population, which is a good thing. So if you have a mutation that's 70% prevalent in the population, there's probably some enhancement to survivability for that organism that has selected that mutation and amplified it through the general population. Just because 70% have it is not a bad thing. And the term mutation, again, we have a colloquial understanding of it, and I think the general person looks at this and goes, oh, I'm a mutant, or I have this poor thing. That's not the case at all. You just have a variant of this. It could be increasing the throughput of MTHFR. It could be increasing the throughput of methionine synthase. What's really important is so just to say that 40% of the population has an MTHFR mutation, it's a big fat so what?
Yeah.
Yeah.
Well, how often are mutations actually a beneficial thing? Because I've heard too that mutations are generally a negative in the sense that they don't generally produce a positive outcome.
Absolutely not. In fact, more often than not, the mutations that we have in the general population across all mutations possible are beneficial. Think about it. If a mutation is bad, what are the likelihood of that person surviving? It goes down, right? It is less likely to be selected in the next generation. So over time, mutations that are still existing and still propagating in the general population are the beneficial ones. So the more we evolve, the more these mutations are becoming beneficial to us. This goes back to, first principles of evolutionary theory. This mutation word has just gone so out of hand, and they think that they're sick or they have this serious ailments because they have a mutation. Absolutely not. You can have a mutation that's beneficial. And when I talk to customers who come to me with this, sort of MTHFR gene that hangs over their head like a sword of Damocles, I have to talk with them and say, do you know which kind of variation you have, what mutation you have? No, it's just a mutation. It doesn't make sense. It doesn't make sense that somebody's been telling them that, no, this is the end all be all. You have this thing, you're sick, or you will be sick, or you're at risk of sickness. And they have no idea of how it's affecting the function of the enzyme that gene codes for.
Yeah, that was another thing I was going to ask. So if we identify a mutation, how do we identify the outcome that has? Because if we don't have good, like if everybody has a slightly different mutation too, we're not all getting the same mutation, I would imagine. Then it would be very difficult to extrapolate that to what the outcome is going to be. Right.
Exactly.
Because a lot of this genetic testing is based on what other people, like 70% of people with cancer have this gene, this gene, this gene, this gene. And so therefore you have those genes too, or that expression or mutation or whatever. And then therefore you have a higher likelihood of cancer. So it's all like basically benchmarking against general population.
Absolutely. That's correct. So we want to talk about methylation, right? And so we know that methylation is this addition of a methyl group, right? And so let's talk about some of the other things that we have, right? So for instance, what we see here is the percentage of total biochemical reactions in the body, rounded to a nice round number roughly, that this type of reaction accounts for. So 1 to 5% of your body's total biochemical reactions, every single reaction that takes place in the body, only 1 to 5% of it is methylation, is adding or removing methyl groups, okay? 1 to 5%. as you can see, it is the least important of all the chemical reactions that do take place. And we'll explain what all of these are. And then on the bottom, on the x-axis here, we have the number of genes that we know of currently that regulate that process. So if methylation, the least important of all the processes that take place in your body, there are 200 genes that regulate it. Now you could have a, very poor methylator, so you're looking at more like on the 1% of the end of the spectrum, or it could be a very high methylator. Your rate of methylation is very high relatively to your other processes. So you could have 5% of your total bodily processes regulated by methylation. And there's 200 genes that we know of, again, that account for that, right? So let's talk about, like, for instance, redox reactions. This involves the transfer of electrons between molecules. This accounts for 10 to 20%. It's highly important in cellular respiration. It is, so think TCA cycle, think electron transport chain, right? These are very important reactions. 10 to 20% of your overall system's chemical reactions are redox reactions, and more than 1,400 genes contribute to the rate of that redox reaction. You have two different types of reactions here. You have hydration and dehydration, and then hydrolysis and condensation. So the latter accounts for 5 to 15% of your total molecules. And so you have, this is important for various pathways. So when we talk about hydrolysis and condensation, hydrolysis is a reaction whereby a water molecule is added to break apart a bond. And then we have condensation synthesis, whereby the water molecule is removed, therefore synthesizing a bond. That accounts for 5 to 15%. And there's 800 genes that regulate that. Phosphorylation, the adding of a phosphate group. This is really important when we talk about energy transfer, for instance, ATP generation and removal, as well as cellular signaling, right? Glucose is phosphorylated and trapped inside cells. Those are phosphorylation reactions. There are people who will tell you there's no molecule in the body that doesn't get methylated. That is a lie. That is a bold-faced lie. There are plenty of other types of reactions, and if we look at the scientific pedagogy to see roughly which ones are the most important, methylation comes dead last. Okay, and so
So can you define how you're saying important? Because you're saying least
number
important,
of
most
reactions.
important, but it's based on amount of reactions. If you were to put that against like outcomes, let's say in terms of diseases, like the likelihood of generating a disease, would those weight roughly the same, or what would the difference be?
Yeah, it might change a little bit, especially when we talk about methylation's role in DNA expression, but it won't change much when we talk about, hydrolysis, condensate, or hydration, dehydration, which is similar, or you might think it's similar to hydrolysis and condensation, but it's not forming or breaking apart a bond. It's just adding a water group, right? Or removing a water group. 2 to 7% between 300 to 400 genes are regulating that process. That is important for chemical digestion. You would not be able to digest your food without that. Ligation is a more important process when it comes to DNA expression than methylation is by far. 3 to 8% of your total chemical processes are ligation, and 400 genes account for that. Okay, isomerization. If you are poorly creating isomers, which are two molecules that have the same chemical structure, but they have a different formation in three-dimensional space. Those are 2 to 7% of reactions, and they can cause debilitating disorders. They can, if you have a isomerization, it can cause even adult mental retardation. It can cause developmental disorders. It can cause issues with fetal development in utero. We have transamination, deamination, adding an amine group or sort of changing that around. This is crucial in amino acid metabolism. This is crucial, proteolysis. How are you going to digest proteins without proteolytic conversions? 10 to 20% of all your processes are proteolysis. About 1,000 genes regulate that. Glycosylation. So this is really important in protein folding. If your proteins do, so glycosylation is when we add a sugar group, a sugar moiety to a molecule. That is really important in folding the proteins in the right way. So let's say you have no mutations. You're perfectly unmutated person, and you create all this RNA from that perfect DNA, and then you get to, you wouldn't have perfect mutations because then obviously you'd have mutations during glycosylation pathways. You get to the RNA. The RNAs are being transcribed into these, single, protein sheets, and then we're trying to fold these proteins because remember, with proteins, function follows form. So if they're not folded correctly, if they don't take appropriate conformation, what happens is the protein doesn't work. Proteins are the basic functional unit of the body. 5 to 10% of all your reactions are with respect to protein folding. If these aren't folding, who cares if your DNA is methylated or not? You're not going to survive. You're not going to be able to do basic things. You're not going to see. You're not going to hear. You're not going to have tactile responses. So when we talk about the importance of methylation, again, you are more than the metabolism of a few micronutrients. You are more than the metabolism of estrogen or neurotransmitters. In fact, the body is relatively robust, and you would be shocked at what it can endure, even on a biochemical level, and still maintain, right? But when we talk about removing your proteolysis, your hydration, dehydration, glycosylation, your redox reactions, if you have an impairment in that, your mitochondrial cycles aren't turning. You have an energy deficit. If you are not engaging in appropriate glycosylation, you're not going to have proteins that are folded correctly. Your body won't function. You will not last minutes, right? If you're, let's say, all of your glycosylation pathways.
But those people probably aren't getting genetic tests either. If they're only alive for a few minutes.
Yeah,
so
exactly.
That's kind of why I'm trying to wait out, so we have the percentages here and that kind of thing, but what is the practical outcome? I think at the very least, what I'm grabbing from this, and much more than that, but in the least sense, is that we need to have a much more holistic vantage point of what's going on from a genetic expression and mutation perspective to be able to understand, how are we assessing the body and its needs, because if we're just looking at methylation, we may not be addressing some of the other deficits that are there that can be supplemented with, that can be mitigated.
Exactly. And I would advise anybody who's looking to get their genetic test done, do not opt for these 4 methylation genes, because again, can they give you some important perspectives? Yeah, into some sort of discrete and some integrated pathways, as we mentioned, with folate, B12, and homocysteine. But overall, you're getting such a minor look into your genes. So if you're getting genetic testing, opt for whole genome sequencing. It's roughly twice the cost of these, big 4 genetic tests. And there's many entities out there offering these big 4 genetic tests.
Can you make it easier on people and say one that you and we'll do genetic testing in the future, which I think will be phenomenal based on what we're building out right now, and it'll have so much capability of being layered into the microbiome and the metabolome and the proteome. But as of now, so you're saying don't go get this one, but would there be one that you personally would trust that you say, yeah, this is worth, if you're considering genetic testing, or somebody referred you, a physician referred you to genetic testing, this is a reasonable test?
Honestly, I don't know off the top of my head. We have, whole genome sequencing capabilities here at the core facilities of ASU. If I, for my research, ever needed to do that,
you'll go down to the lab and sequence
exactly,
it, right?
Exactly. Yeah. So I don't know.
Most people don't have a lab in their backyard.
Right. Yeah. So if you don't have a lab 10 feet away from you, then what I would do is I would do a deep dive into a company that is run by scientists. I would look for WGS, whole genome sequencing. Usually these are utilizing next generation sequencing platforms. And I would look for not just how much data they're going to give you, but how do they interpret that data? So make sure that they're giving you some sort of actionable next steps, because if they just send you a file that's like, here's your DNA, what is the average person going to do with that? And so make sure that there is some bioinformatic processing on the back end that will give you some actionable insights into, you have this kind of variation in these important genes, and there will be a list of hundreds of them that they'll most likely give you. And this is what you should do about them. This is how you should modify your life or your diet or supplementation to ameliorate any potential problems. And again, if you have this gene, let's say you have a perfectly good MTHFR and somebody reads it and it goes, no worries there. Well, there's no guarantee that that gene is not being transcribed correctly to RNA, or that RNA is being translated correctly to an adequately folded protein, right? So why are you even doing this test, right? So when we talk about genetic testing, it should really only be done to refine lower levels of systems biology, which we'll get into.
I think that's an important point, because
right.
Genetic testing has kind of become one of the forefronts of testing, at least with like functional physicians and RDs and that kind of thing, as they're looking at genetic testing as a forefront. But can you explain further on why?
Right. So,
Why
okay,
it's good to have the other layers first?
Right. So let's say, so when we look at the metabolome, right, we can look at the expression of everything upstream. So we're looking at genetic expression. We are looking necessarily at the transcription of genome to transcriptome. We are looking at the translation of transcriptome or RNAs to proteins. We are looking at the interaction of proteins to the microbiome, and then the microbiome interactions with the proteome and the metabolome and everything upstream of it as well. So by testing the metabolome, by testing this lower level, as you can see, the metabolome is ampliative. It contains all of the information of the other ones, roughly speaking, and not only contains information as to what genetic mutations you probably have or don't have, but it's looking at the functional output of those genetic mutations. Again,
Yeah, it's like regardless of your genetic expression, regardless of your transcriptome, your proteome, this is the outcome. This is
the outcome happening
and so we're
right
looking
now.
at the outcome saying like, okay, so we know these levels are off, and we know that if there's validated methods to increase those. And so not that genetic expression or genetic testing is totally out the window, but that we can look at the outcome and say like, how do we feed that to improve that? That's going to have a really quick response, and then go into deeper levels of this person's biology and understand it. I mean, that's how I kind of understand this testing paradigm.
Right. So let's say you have, no MTHFR mutation. So that means we should see high levels of 5-methyl-THF, right? But when we check your metabolome, we don't see that. So that's telling us if we have some genomic data on you on your MTHFR mutation, and we have metabolomic data on you, especially on levels of 5,10-methylene THF and 5-methyl-THF, and we see that the opposite is happening, that you have no known mutation, but that you have very high levels of 5,10-methylene THF, very low levels of 5-methyl-THF, again, the product of this enzyme that the MTHFR gene codes for. What that's telling us is that the process doesn't match up with your genome. So what does that tell us? That means that there's something going on with your transcriptome. That gene is not being read properly, right? Having your genetic test just means that we read your gene using, biochemical, sort of biotechnological means, and we read that sequence and we're like, great. But just because we read it doesn't mean your body is going to read it the same way and create RNA from it adequately, or that RNA is then going to go to your proteome and be translated by ribosomal machinery to functioning proteins, or the enzyme that is responsible for that reaction, or that maybe there's something in your microbiome that is taking your folic acid and consuming it too much, and is producing maybe or not consuming it enough, or something like that, and it's adding to this sort of backup in the molecular biochemistry between the product and substrate that enzyme is responsible for. So you can get this test. You can say, I have an MTHFR mutation. But unless you look at if that mutation is in fact being transcribed and translated properly, interacting with all the other biochemistry in your body, and is producing levels of the product and substrate in a relative range that is in accordance with what we would say this person has no mutation, you don't know. You don't know. The genome is a big what if. And I have no mutation NTR, but what if my transcriptome is messed up? What if my proteome is not functioning properly? What if my microbiome is causing aberrations? I don't know. What I need to test is the metabolome, which is a direct functional readout of all these levels above it. So if you are going to test, well, now ideally, our goal is to introduce tests for every level of this hierarchy. And when we have this deep molecular profile, when we have molecularly fingerprinted their central dogma of biology, again, from the expression of nuclear DNA down through low molecular weight products, which are the metabolites, then we've sort of been able to ask and answer everything, right? Right now, if you had to pick one level to test and one assay, pick a metabolomics test. That is going to tell you where you are right now, and it is going to be the function of all of these levels upstream.
Yeah, we have other videos on our channel that go into this in depth. That's a pretty profound statement that you made too, because I don't want to just gloss over that, because we're talking about multiomics integration, which to my understanding, there is no test that goes direct to consumer, direct to physician that's currently available where you can integrate all these layers of biology and get a really in-depth understanding. I mean, they're getting down to like the scary accurate thing. Like you were riding your bike when you were 6, fell off and scraped your knee October 6, 1972. Like getting, but not for the point of just being accurate, but for the point of how do we make functional changes in somebody's body? How do we resolve some of the chronic issues that are happening? How do we mitigate the biggest factors of mortality in our society? And so it's really getting down to some fundamental things that'll have a very large impact. And that's why I'm a part of this project, is because I believe in that mission and that vision of what we're doing. And that's going to have like a drastic outcome in terms of people's their lives, I've talked about before, but I'm just very excited for this because, perhaps that means that grandparents will be around for their grandkids when they weren't otherwise, or parents or brothers or friends or whoever, right? Like this can have immensely profound impact in society. But, and to go to the other point, so switching gears a little bit, this is going to kind of bastardize what you said in a way oversimplified form. But I think with the metabolome testing, let's say you have two people. One guy, 6-pack abs, the image of a specimen, right? And another guy, it's a dad bod. So the guy with the dad bod, you can say, and you're a trainer, right? You're bringing them into your facility. And the guy with the dad bod, you can pretty accurately say like, hey, I don't know what you've been doing, but whatever that thing that you've been doing is, or whatever you've been doing hasn't been clearly working to get the result that you want, which is 6-pack abs, right? And so you can look at that person and say, okay, I don't exactly understand every mechanism that caused you to get here, but I know what to do about it now. I know you need to sleep more. I know you need to exercise more. I know you need to eat this food and this food and this food, these macronutrients, to get you to your objective of having a 6-pack. And on the other side, we can look at somebody with a 6-pack and say like, okay, I don't know all of what you did to get here, but here is how we maintain that or optimize that even further. And so that's what the metabolome is doing. It's essentially saying, regardless of all the stuff above that and what you've done to get here, here's what we can do to give you the best possible outcome currently in the context of the metabolism, right? That's not going to necessarily change the genetic expression, but that'll mitigate some of the issues that are upstream there.
Exactly. You're totally right. And so what is our test, right? This is the Aristotle test again, and this is a trademark name of what's known as GCMS aqueous profiling, where we take a dry blood spot. It's a direct-to-consumer at-home kit that enables remote patient monitoring. You collect, it's not a drop of blood, but a few drops of blood on 2 different circles. So it gives us roughly 20 microliters of sample, which is exactly what we need to get a very good signal on GCMS analyses. And I invite individuals who are looking at this presentation to go look at some of my publications and other publications in the field that use a similar sample quantity. I think a lot of people are shocked at the volume, but it's old news in science, right? That for especially GCMS, we're running in this mode called splitless mode, which injects the entire analyte without diffusion from the injector site. And so the inlet. And so we are able to get fantastic noises. We separate these metabolites along 2 dimensions. One is retention time, and the other is mass to charge, or the weight of the molecule, roughly. Okay. And then what we are looking at isn't 4 variations of 4 genes. We're looking at 126 metabolites in central carbon metabolism, and they're indicative of 68% of all cellular functions. So it is a very comprehensive, sensitive, and specific, and therefore accurate health assessment. And so this provides you with a much greater view of the functioning of your metabolism. And we can compare that between, let's say, a methylation test compared to our Aristotle test. So when we say methylation gene test, we're talking about, those 4 that we covered, the ones that, a dozen or so companies are right now offering to you and making, again, much ado about ultimately nothing. So,
and they're not clear about it, by the way. Like if somebody does whole genome sequencing, they're clear about it because it's something to stand on to say we do whole genomes. It's just like if somebody has like organic, they're not going to, they're going to put that on the label because it's going to get more buyers.
Exactly. If you go to, one of the large companies that does this, and you look at their, website, they don't even say which genes they're testing for. They just say that, they mention MTHFR, it's the only one. And then they say 40 something percent of people have this mutation. And again, they're preying on this fear, this misunderstanding of what mutation is and how it affects health. I sometimes wonder if they themselves understand it. But
well, that begs the question then, when would you consider doing genetic testing? So if you are trying to understand how somebody's biology is functioning and what you can do to get them healthier, when in this pipeline would you consider genetic testing? Like, what would you be looking at?
Yeah, I would look at
you to that?
Yeah, I would look at the genetics basically at the end, right? If I can test your metabolism and I see that there's some issues with, let's say, alanine aspartate, I might then go and recommend you either do a proteomics panel, like an enzymatic proteomics panel with some sort of tryptic digestion, or I would recommend you to just go do a functional liver test at your local lab. Go get your ALT and AST done. And then that tells us, the activity, the function of those enzymes that are responsible for alanine aspartate, right? So we would go step by step, but looking at the cellular, intercellular, extracellular milieu is going to be the first step in seeing where you are. And ideally from a sample type that is systemic, like blood, right? A lot of people say, why not urine? Well, urine's an excretory profile. It's basically all these metabolites that have been marked for excretion. They're not in their active functional forms. It doesn't do us any good. Or feces really just speaks to intestinal health from the alimentary canal. Or if you get CSF is great. First of all, there's no at-home spinal tap, so remote patient monitoring goes right out the window. And then also, but if you're looking at something like Alzheimer's disease or Parkinson's, CSF is the most desirable tissue type, aside from postmortem autopsy of cortical tissues. So when we look at something like canonical KEGG pathways, KEGG is the Kyoto Encyclopedia of Genes and Genomes. It is the database that is sort of standardized when we look at all disciplines of biology. There are other databases that are more specific to metabolomics, but KEGG is everything from genomics to proteomics to metabolomics. We utilize the KEGG database, and they have put in, various pathways that are responsible for human biology. And when we look at the 19 substrates and products that those 4 genes and their variants account for, and we look at the 126 metabolites of the Aristotle test, we see that these methylation genes are looking at 5 biochemical pathways in total. 5. In comparison, our 126 sentinel metabolites are indicative of 80 different biochemical pathways. That same ampliative nature of a metabolomics panel is expressed with every other category. When we look at human disease signatures, those 5 genes representing 19 substrates and products are indicative of 42 human disease signatures. In contrast, our test is able to look at 480 validated disease signatures in human blood. Now, you might see that, okay, why does 42 and 480, why is 42 more than half the size of 480? Well, that's because we had to change the x-axis here because some of these are so high that we wouldn't be able to see the others. So, these are, in exponential base 10 units here. So 42 versus 480. So it's not that these aren't important, right? The genetic methylation test, you're going to get 5 pathways. You're going to get 42 potential disease signatures or propensities. Really, they're not even signatures. They're just what ifs, right? They're likelihoods, right? But whereas we can say, no, you have a 33% overlap with the metabolic profile of Alzheimer's, right? We're looking at what is, not what could be. And then when we look at SNPs or single nucleotide polymorphisms, these are the most common types of genetic mutations. Necessarily, they're looking at 4 of them. We are able to profile 4,598 of them. So this is again how the metabolism is used as a direct functional readout of the genome. You may have these genes, you may not, but let's look at your metabolism to see what's being expressed. And given the levels of those metabolites downstream, we can infer the presence or absence of 4,598 mutations. When we look at enzymes, they're necessarily, again, looking at 4 enzymes, right? The transferase enzymes, MTHFR, methionine synthase, and adenosyl homocysteinase. We are looking at the dysfunction in 912 enzymes. when we look at how much are these enzymes, right? If we looked at how many of these, tissue types, so when we look at tissues, organs, or subcellular organelles, right? When we talk about ribosomes, mitochondria, when we look at liver tissue, when we look at cardiac muscle, etc. Out of all the tissue types and locations and subcellular organelles and everything, how many of those locations are expressing these 4 genes? 34. So they're pretty prevalent, actually, right? Like ComT, MTHFR, these are present in 34 different tissues or organs or subcellular organelles. Conversely, our 126 metabolite panel is indicative of 78 different tissues or organs or subcellular organelles. So while they can speak to 5 pathways, 42 disease signatures, some genetic mutations, some enzymes, and 34 tissue sites, localizations, we say, in the human body, we have 80 pathways, we have 480 disease signatures, we have almost 4,600 different mutations we can profile and do profile. We have 912 enzymes we can check for dysfunction, and we have 78 of more than twice as many ubiquitous localizations than these methylation tests.
I think there's been a lot of trust destroyed in laboratory testing because I think there's not, it seems like, there's some things that physicians can utilize that give them good indication of what's going on, but when they're not sure what's going on, they'll pull a lab and say, hey, your labs are normal, and the person feels very, very sick or something like that. And so I think there's been a lot of trust destroyed in this place, which gives a platform for people to offer all sorts of testing on all sorts of things that may not necessarily be the thing that is needed. But I think that's how that, we get rise to a lot of these organizations that are offering partial testing and different things, because people are really trying to get to the bottom of their health state and understanding what they can do about it. Or on the flip side, people are becoming very interested with longevity and what they can do on that side of things. But I think the typical labs that are used, there's just, when you hear so often that, I'm really sick, and then it comes back, labs are normal, I don't know what's wrong with you. I think people, start to look elsewhere.
Yeah, absolutely. And that's because traditional diagnostics, when we compare them to molecular diagnostics, they're lacking in sensitivity. So they are lagging behind. If you tested that same 6 person, then maybe 4 to 5 years later, the lab results will, I think, be more abnormal. But when we're looking at, very in-depth peek at, cellular biochemistry, we're able to monitor and detect very slight changes that often precede other changes in traditional diagnostics by half a decade, if not more. So,
yeah, and there's a lot of people that understand this psychology too, that once you have third party, which is also why I'm a part of Therium. I love that we don't sell supplements, and we will never make $1 from supplementation or the intervention they do afterwards. Separating the bias between what they do after the test and the test itself, I think, is very important. If there is a clear link there, I was talking with somebody who works with an executive at one of these companies, and he was really just dismayed because he was having a conversation with this person and asking about how they do their customizing their supplementation plan and all that kind of stuff. And the executive said, "Oh, well, I stopped thinking about supplementation a long time ago. It's all about data."
Yeah,
and so I think there's an opportunity now that we've seen, like, okay, typical lab tests have a place. They're not giving us the whole picture. There's all these private companies that are being started, going direct to consumer and that kind of thing. And some of those do have a place, and they do good testing. But then there's translation, interpretation. What does this mean? What is the weight of this thing? And so that's what I love we're doing at Therium, is really kind of building out the template for how testing should be done in the future, such as, we've talked about building this onto blockchain so that the consumer owns their own health data. They can share in the profits, and they consent each and every time to a sale of data very transparently. And it's not just kind of this one-time consent and then it's blind from there. It's each and every time, or just the fact that where we don't have any supplementation or any bias on the intervention. And I think that gives a platform for really radical change. So I'm super excited about it.
Yeah, exactly. And we'll get into the supplementation, and we're going to get into digital twinning now, which leads into the sort of our agnostic approach to supplementation, and all the things. We're not doing health coaching, we're not selling diet plans, and so we'll talk a little bit about digital twinning. And I've talked about this before. And what we're looking at is, okay, now we have all of this data. What do we do with it? It's more than just saying, you need more folic acid, or you need to take a methylated version of B12. It's far more than these sort of single point interventions to solve what are undoubtedly not single point problems. So we have, this concept of digital twinning. Very briefly, I don't want to get too much into this because we have so much more information on this in our other webinars, but it is a concept that was borrowed from the engineering and aeronautical industry, whereby a thing, a desired state, is designed in silico, tested in silico, and then in the context of engineering, that thing is only fabricated or processed once. Let's talk about a fuselage of a spacecraft, for instance. This is a tool that NASA developed, a few scientists at NASA. In the last 5, maybe 6 years, it has had growing adoption and appreciation in the health sciences. And it's still in its very nascent form, even in academia. It's still in its very nascent form, and I'm very pleased to say that Therium is sort of one of the leaders of this vanguard in terms of the research we're doing and the publications we're getting out regarding digital twins. It is a tool that enables doctors or researchers like myself to visualize the complex physiological processes and then simulate various interventions on that individual. So we can say, let's take niacin and change 15% of the parameters of niacin supplementation. So the dosage, the frequency, the time of day, what meals you consume it with. Let's change the parameters and iterate that 1,000 times. So we are testing niacin supplementation 1,000 times on the digital version of your metabolism. And we're viewing the response of the metabolism across those 80 pathways, across those 912 enzymes, across those 480 disease signatures. And we're seeing which one gives the best outcome. And we're doing this now for roughly 1,300 interventions, everything that ranges from metformin therapy to over-the-counter supplementation to chelation therapy, infrared sauna, even aspects of health that are only now growing in appreciation, but for which we have long-standing biopsychosocial data. For instance, social connections. We know that this, in the behavioral sciences, is a very important part of mental well-being and longevity and quality of life. And so we can say things like, hey, interact with social groups more, or learn a brain training game to increase neuronal dendritic density, right? The connections between neurons and how many connections are formed. So it serves as this predictive tool. And we take, your metabolism, and then in silico, we model all of those interventions, again, each 1,000 times with 15% of the possible parameters changed. And what we're doing is we're saying, okay, this is akin to you going out and trying 1,300 different dietary, exercise, lifestyle, supplementation, etc., medication regimens, and trying each of those 1,300 different things out 1,000 times in different ways and figuring out what's best for you. Can you imagine the time and the cost? You might kill yourself doing this. What we can do is say, okay, given the metabolomics data that we have, and given the relevant biopsychosocial data that you report, we can simulate on that metabolism the probable effects of what we're seeing, mined from the literature, all with scientific studies to corroborate them, and we can assign these weights to them and see your metabolism's response, and then collate a protocol, very personalized, complementary, customized protocol for you to sort of shore up this health gap. So this health gap itself informs what we recommend, and so through time, this process becomes more refined, especially as we ascertain how you respond. Because at the first test, we can only predict how you will respond. At the second test, we now have a measure of how you actually responded, right? And then that actual measure, that delta score, becomes more refined through time, and we're able to give you more personalized, more efficacious, personally efficacious, treatments to optimize your health outcomes. And as you mentioned, Micah, we don't sell anything. We don't sell a diet plan. We don't sell supplements. We don't sell an exercise routine. You will never see me sell me on DVD and doing an exercise routine whatsoever.
6-minute
especially
abs.
Me. 6-minute abs from Dr. Jasbi. No, I'm the dad bod guy. So yeah, it's fine. But so this is not something that we've it's incredibly disingenuous for a company to sell you a test with the right hand, sell you the supposed solution with the left. And I did that actually opposite. But it is incredibly insincere. And what we are doing is a genuine scientific effort to match you with what your metabolism needs best.
Yeah, it's very exciting technology, and it has immense potential to grow. And I know you're on limited time here, so I'll let you get back to it. But I think there's a lot more discussion we can have too in the context of not only genetics but metabolism and the other layers of systems biology. I think it would be really cool to have a conversation that kind of maps all that out in a way that's really practical for physicians and consumers that are interested too, of course.
Absolutely. And so we will talk about, and we're talking about some case studies, and you can go to our website and download the electronic version of this book we have published as a diagnostic hand guide, as a handbook to guide both direct-to-consumer individuals as well as clinicians and physicians on the use of this test. So I've pulled some of the case studies, and these come from the early days of our beta testing. And some of the case studies, as you'll see, are direct-to-consumer individuals. Some we worked with physicians. When we work with physicians, we're able to then give more prescription recommendations. We're able to say, okay, you have a probable signature of heavy metal toxicity. You reported being a retired dentist. It's most likely mercury handling dental amalgams, et cetera. We're going to give you chelation therapy. And that physician who's working with us can prescribe that. Otherwise, we have to resort to things like sulforaphane through diet or supplementation, increased hydration, increased aerobic activity, infrared sauna sessions for a certain amount of time, for a certain frequency during the week. And so we're going to discuss some of these. This was very early on. We had Anita. She was in remission for breast cancer. She had persistent fatigue. She had persistent neuropathy. Now, these are common symptoms of somebody who has undergone chemotherapy. And what we were able to restore in her was her vitality. The neuropathy we weren't able to address, in all honesty. It was the fatigue that we were seeing. And by looking at her mitochondrial metabolites, so we monitor 5 of the 8 mitochondrial products, in addition to adenosine, which we can use to infer the levels of ATP, we were able to see this characteristic arrest of her mitochondrial metabolism. We were able to give her a very antioxidant-rich diet, mitochondrial support supplements, including CoQ10 and PQQ, at very prescribed, personalized dosages, a little bit above what is the standard recommendation on the back of the bottle, sort of back of the bottle recommendations for these. And over 3 months, she reported, she did not retest with us, but she reported a return to her pre-illness energy levels. And so that is one way that we were able to take Anita, deconstruct her, to perform this mathematical abstraction of her metabolism to her digital twin, simulate all these interventions, and come up with a way that we have been able to boost her energy levels by addressing, again, the high oxidative stress load, as well as this arrest of her TCA cycle that we were seeing. We had Jake, 45-year-old teacher from the East Coast. He had just, again, persistent fatigue, cognitive difficulties. These are things that we all experience from time to time, right? But he had tried traditional diagnostics, looking at, complete metabolic panel, which is anything but complete, thyroid tests, neurological evaluations by his physician. His physician was who actually prescribed him referred him over to us and introduced our testing. So his results actually showed this characteristic deficiency in his purine metabolism, which is important for processes related to energy production and DNA repair, et cetera. And he also had one particular elevated stress marker. His physician, who was an incredibly well-trained physician, who, again, is one of those who is leading this push to formalize personalized medicine as a distinct medical specialty. He diagnosed him with this very rare IEM, inborn error of metabolism, related to his purine synthesis. And so we changed his diet around. We were able to simulate various dietary interventions to find what is best with him. And his physician, recognizing his deficiency in purine synthesis, was able to prescribe him oral enzyme supplementation in order to, since he wasn't expressing these enzymes, to take them in and to create more purine metabolites as a result. So he saw a considerable improvement in his energy levels and his cognitive clarity. And this was within 2 months upon receiving his results and consulting with his physician. And then finally, we have Rohan. Yeah, do you have a question, Micah?
Oh no, I mean, yeah, these case studies are wonderful. I mean, just so we're a small company, right? I mean, we're doing some amazing work, but I'm still helping people, answering their questions and all sorts of stuff, and I love to do it. You know, if you know me with some of my other businesses and things that I've done, I find that there's so much value and reward in assisting people with all sorts of questions and kind of being on the front lines to a degree. But yeah, I've just had lots of tremendous feedback from people over the past year in terms of being like, oh, this gave me exactly what I needed, or gave insights. You know, unfortunately with HIPAA compliance and all that, I wish I could share more, but even I had a conversation with a gentleman that went way over what I anticipated. It was supposed to be 30 minutes, ended up being like 2 hours. And he had a son with cancer.
Hello,
and he said he had spent an additional $50,000 outside of what the physicians ordered for testing on additional tests. And
Yeah.
he said this was one of the most impactful in their journey. So it's been pretty cool.
Yeah. We've seen a lot of improvements, both colloquial as well as people who have retested with us and seen, oh, if I did their recommendations, if I took this supplement and I ate and changed my diet this way and changed my microbiota in this way, I did feel better, but I also tested better. So there's this quantitative substantiation that often complements their own sense of well-being. And then finally, I'll end here with Rohan. Recurrent respiratory infections, gosh, I had them myself when I was a kid. And so he was getting them too much. And it started to really become too frequent, from once a year to twice a year to 3-4 times a year. We saw that there was a distinct change in his lipid metabolism, particularly levels of palmitic acid, which is an aryl hydrocarbon receptor, which can actually modulate the immune system in very negative and detrimental ways. And so we, again, were working with his immunologist, and we identified that there was this primary immunodeficiency that was related to his lipid metabolism. So we changed his diet, again, going to our digital twinning platform to find what was best for him. Reducing, for instance, his use of coconut oil was through the roof. And coconut oil is replete with palmitic acid. And so we tailored that out, brought in some healthier fats, evened out his lipid profile, palmitic acid, linoleic acid, alpha-linolenic acid, oleic acid, etc. And we're able to work with his physician to prescribe a very tailored dose of immunoglobulin therapies. So these are antibodies secreted by B cells, and we can give them as therapies. And of course, his immune resilience improved dramatically, and he is free of these recurrent respiratory infections now. And so just briefly want to talk about what we've got coming up in our pipelines. Very exciting time at Therium. We are developing the Ilia gut microbiome test, set to launch in summer of this year. This is a metatranscriptomic testing platform. This will give us 600 species of bacteria, viruses, archaea, fungi, even parasites, and integrating that microbiome data with the functional metabolomic data. We are set to provide our customers with unparalleled insights and offer the first truly multiomic test on the market. And this goes far and above what traditional gut tests are, which are 16S tests. So they have very poor species level resolution and often now are not just old but obsolete. We also are launching a mobile health app. This is going to just enhance the Therium customer experience. You'll be able to order testing, schedule consults, schedule a time for my own office hours. You'll have streamlined results interpretation. And then with integration with wearable data, we'll be able to add another layer of data that is not systems biology related, but behaviorally related, which is also incredibly important in moving the needle on an individual's health. And finally, as Micah mentioned, we will be implementing a blockchain. So this is a cutting-edge security technology. So you give us the rights to your data when you take our test. We do not sell this data, not to academic collaborators, not to pharmaceutical companies, not to life insurance companies. It has never been sold, and it never will be sold. Our goal is to implement this, encrypt this on a blockchain network. Once we encrypt this on this blockchain, it will be a transparent, trustless domain whereby every single request to access your data will come along with a smart contract, which will enable you to know who's accessing your data, for how long, what portion of your data, what's it being used for. And because it's smart contract enabled and access tokens are granted in exchange for retrieving your data, you will actually share in the profits. So this is one way that we will incentivize our customers to share their data, because data sharing to reputable sources is the backbone of the scientific enterprise. It truly is important for making scientific advances. We need to make sure that whatever data storage and sharing software we implement as a company, that it still allows for data sharing and academic collaborations and scientific collaborations without intruding on or impeding a person's right to their own data, to know when their data is being sold, to who, for how long, for what purpose. And of course, it's their data. So it makes sense that they should be able to share in the benefits as well.
And hopefully we see some mass adoption in this, because as far as I know, we're the first ones that have conceptualized this and not only conceptualize it, but actually have a plan in place that is able to be attained. And so I think this should be the standard. Because the only reason I haven't taken like a 23andMe is because they're going to sell my data to the government and to Pfizer and whoever wants it, essentially. But with this, you essentially have the vault and the key. So it's only accessed with your permission each and every time. And so I think this should be the standard of health data as far as if it's ever utilized for other entities outside of the one that you tested with.
Absolutely. And so there are, of course, academic papers. Ours, of course, are part of those that we've published on with the blockchain and theorizing how this could work. There is a vanguard leading implementation of blockchain in healthcare. I can't say that we're the first people to think of it, but we are the first company to implement it. And as you mentioned, yes, this should be the standard going forward. And so if you need any more information, please, Micah has been kind enough to put a discount code for all of our Aristotle testing tiers. 10% off all non-subscription testing can be achieved using the code SPRING. That expires Sunday, April 7th. If you have any more questions, please do not hesitate to give us a call. Do not hesitate to email us at info@therio.me. Go to our website, download the Aristotle handbook, and familiarize yourself with systems biology, metabolomics, and what we are doing to make strides in those domains. We are located here in the beautiful Wexford Science and Technology building here in downtown Phoenix. If you're ever in town and you'd like to schedule a lab tour, email info@therio.me maybe a week in advance, and we'll make sure that we give you guys a tour of our facilities. Also, you can book an office hours time with me by navigating to therio.me/questions. This is not a link for individuals who want to discuss their test results. That's a separate link, which you'll get once you test with us. And if you select the consult, you receive the link to schedule one. But it's a very easy calendar link that you can schedule here just to get more answers to questions you might have. I'm sure this presentation has drummed up more questions maybe than it's answered, which should be a good presentation then. If you have any questions, please book my office hours at that link, and I'm happy to sit down in 30-minute intervals. A few times a week, I make myself available to discuss with physicians, clinician owners, as well as direct-to-consumer persons. So if you're not a physician, you just want to get more information, please don't hesitate to reach out to us.
So thank you so much, Dr. Jasbi. It's been absolutely wonderful to speak with you about what I've been curious about, which is why are so many companies testing for these 4 methylation genes, and what impact do those have on the body? I think there's more discussion to be had in understanding not only genetic testing, but the other layers of systems biology, how those can be tested. Because what we're trying to get to is like, how can we have actionable insight into what's happening? Because I think there's a lot of different tests that people can do, but I think we're roping it together and we're bringing it and consolidating it into one spot where people can really understand their biology. But that takes time. And right now we do have the metabolome, which bar none, I think is the best test out there. But I think there is opportunity for further discussion here where we can get into maybe some of the details and the practical insights of how a physician or clinician RD might utilize some of these different layers of testing. So again, too, if you're interested in getting a test and understanding your biology and what you can do about it, maybe you have the dad bod and you want to go to the six-pack abs as a metaphor. We're talking metabolically, like what's happening inside your body. Then we do have a discount right now. You can use code SPRING on www.therio.me. That's going to get you access to a test kit. We'll ship it to your house. You collect using a fingerprint prick, put a small drop of blood onto a sample that goes to the lab. We send the report within 2 weeks of receiving it. So that's how that process works. And I thank you guys for joining us today.